oxidation is highly regulated. And this is one of the things I said before, is that scientists have tried to figure out the key components to target, like the carotene acyl transferase number 1 system in regards to the transportation of short-chain fatty acids into the mitochondria, to allow us really to be able to hopefully combat the situation of changing the metabolism within the mitochondria. We haven't been successful because one of the problems is you start changing the metabolism in the mitochondria, which is the powerhouse of the cell, you're going to kill the cell. So, it's been an issue where, we've moved some advancement, but not the advancement of being able to understand the full regulation and as well as the implications on the mitochondria and its existence and function within the cytosol. So we know fatty acid acyl-CoA production, particularly in the mitochondria, occurs through that process we studied yesterday in beta oxidation. We are also going to see outside the mitochondria the processes to start the advancement and promotion and preparation of fatty acids to undertake that process to move into the mitochondria. So with our triacylglycerols as well as our phospholipids, which are present in the cytosol, we're going to see those lipases start breaking those down and trying to get to those smaller short-chain fatty acids, which are compatible to the carotene acyl transferase number 1 enzyme to move across the double membrane of the mitochondria and then to be regenerated into the matrix of the mitochondria as acetyl-CoA fatty acid chain, not as a triacylglycerol. Remember, we can't move those triacylglycerols through. We can only move the shorter chain fatty acids, and they can be saturated or unsaturated because they're going to be processed in the process of beta oxidation in the saturated, unsaturated, mono or trisaturated or disaturated form in regards to the mitochondria. So again, that mitochondria plays a very important role in, central to the oxidation and metabolism of fatty acids. So the fatty acid limitation becomes then the rate of how the carotene acyl transferase number 1 enzyme can handle the free fatty acid as a coenzyme conjugate and then transport it across the inner membrane space of the mitochondria and then actually regenerating it inside the matrix of the mitochondria. So that rate of movement is going to be the rate limiting step in regards to the process of fatty acid acyl-CoA oxidation in the mitochondria. Now, though we don't talk much and illustrate much on fatty acid biosynthesis, one of the first intermediates in fatty acid biosynthesis, which we've already been kind of introduced to, is a manyl-CoA. Remember, we talked about the propanol-CoA and producing a methyl-manyl-CoA. We can produce manyl-CoA from that intermediate and then push it through to fatty acid biosynthesis. Now, what we see with fatty acid biosynthesis is the utilization of the acetyl-CoA, those 2 carbon units, basically being mounted upon an elongating chain of a fatty acid. So again, we have to have that source of acetyl-CoA coming into the mitochondria to be able to produce those longer chain fatty acids. So again, we're going to have that transport system via the carotene transferase number 1 doing its job also to regenerate both CoAsh as well as a source for acetyl-CoA to make those particular fatty acids and then be able to transport those out. So we're going to have a little bit of a look at some of this, but I'm not focusing in this particular course on too much on the fatty acid biosynthesis. That comes in the plant biochemistry course if you so desire to do that. So on chapter 17, pages 661 to 688. Excuse me. A little dry today. So The acetyl-CoA carboxylase is a major uh, regulatory enzyme in regards to the synthesis of fatty acids. And what we see here within the cytosol and the reactivity of the low blood glucose level or the high blood glucose level on the process of phosphorylation of the acetyl-CoA carboxylase, the ACC enzyme. And again, we see a role of the protein kinase A playing a particular component in regards to the deactivation of the AAC by its process of phosphorylation. And again, this is going to be stimulated by a GCPR in response to low blood glucose levels, and that increase in that peptide hormone glucagon interacting with a GCPR and then the adenyl cyclase to allow the protein kinase A to do its phosphorylational job and shutting down the system. What that will do is actually implicate and decrease the amount of manyl-CoA being produced from acetyl-CoA, because it's that manyl-CoA that is going to be channeled into the cytosol to produce fatty acids. The manyl-CoA also has another role in regards to inhibiting the movement of the fatty acids that are being produced in the cytosol, because we have the energy, because we have the acetyl-CoA, in regards to their transportation into the beta-carotene system shuttle to allow them to be further degraded into the acetyl-CoA inside the mitochondria. So again, we always have this particular balance occurring. If we are synthesizing fatty acids, we're going to decrease their transportation into the mitochondria, which promotes their degradation. So that's a logical particular process, as illustrated here with number 4 with the manyl-CoA. So if we have high blood glucose levels and we're secreting our hormone insulin, again, working through the insulin receptor and the insulin substrate number 1, basically going through the cascade of events and activating a phosphatase, which is going to dephosphorylate the acetyl-CoA carboxylase enzyme to make it then become active. With that activity, that acetyl-CoA carboxylase enzyme is going to actually consume acetyl-CoA and produce manyl-CoA. With that manyl-CoA, then we get production of fatty acids, which will then become triacylglycerols, which will then be stored in the lipocytes or in the tissue or in the liver, or be utilized as an energy source at a later stage. And that comes from the issue of dietary carbohydrates, which are going to increase that blood glucose level. But we can also get that glucose level and the degradation of glucose, as we know, from the degradation of glucose through glycolysis to give us that acetyl-CoA through the pyruvate dehydrogenase complex. So not entering the issue of the TCA cycle, because what we remember, that citrate is inhibiting the citrate synthase and other things, that the ATP concentration and the NADH concentration is inhibiting that acetyl-CoA from moving through the pyruvate dehydrogenase complex and entering the commitment stage of the TCA cycle. But what we're seeing is that now acetyl-CoA being shuttled off by the ACC to produce that manyl-CoA to allow us to then produce fatty acids. So, the rule is when we see an increase in glucose, again, the cell is happy, the ATP and the NADH and FADH concentrations are high, the AMP concentrations are low, as well as the NAD plus concentrations are low. The cell is in a happy state. So what we see then is the decrease in beta oxidation due to this inhibition of the transport system. We also keep in check and balance, as I said before, this NADH and Na plus ratio. Now, this particular ratio is sensitive to one of the enzymes that are required in the process of beta oxidation, the beta hydroxyl acyl coenzyme A dehydrogenase. And that is inhibited, that particular process, which basically increases the acetyl CoA concentration in regards to inhibiting the thioester or the thiolase, the last step in breaking the fatty acid to produce more acetyl CoA. Because again, we have a feedback system here. If there is more acetyl CoA in the system than needed, it's going to actually prohibit the concentrations of acetyl CoA are going to prohibit that thiolase enzyme to cleave more acetyl CoA off the chain of fatty acids. So again, hopefully you're starting to come to appreciate every time we regulate a system, there's going to be a regulation of enzymes and there's going to be a regulation of a transporter or transporters to basically minimize the product that is the regulator in regards to providing you this negative feedback. So, as I said, we know some of these dehydrogenase effects because of particular diseases. And some of these diseases are pretty nasty in regards to those who get them basically don't survive, usually beyond childhood. So here we have some genetic defects in the fatty acid acyl CoA dehydrogenase enzymes. They are pretty rare, 1 in 10,000. And the problem is they don't have the ability or the enzymes because of that acyl coenzyme dehydrogenase enzyme mutation, don't have the ability to oxidize free fatty acids of a particular length, this time carbon 6 to carbon 12 length. So the short chain fatty acids that are usually utilized in the transport system to feed the mitochondria. Now, the sad thing is these short chain fatty acids start to accumulate quite rapidly in the neonate, into the baby. And so death is usually 25 to 60% within early childhood. But we also see other things in regards to mental retardation, cardio atrophy, meaning a small heart, muscle development, and as I said, I think mental retardation. And usually, actually, interestingly, some myopic eye or hearing auditory issues in regards to the nerves. Because remember, the nerves have these lipids around them which act as a myelin sheath or a conduct or wire insulator. And because they're not producing those fatty acids with the growing child, those fatty acids, which are normally in the myelin sheath, are actually metabolized as an energy source. So basically, you're stripping off the insulation of your central nervous system, and which causes convulsions and some other issues, which we'll see in some other diseases, because they all follow a very similar pathology. So the human genetic effects of this fatty acid oxidation, or importantly, fatty acid transportation, accounts for about 20 particular diseases. Those particular diseases basically, in most cases, are supplemented, and there's ways the body will actually go around and do other metabolic processes to combat some of these long chain or short chain fatty acids to a level they will start accumulating a byproduct, and this is the byproduct we particularly look for. But majority of those are usually lethal in early childhood. So we'll see, particularly, as I said, heart disease, abnormal skeletal development, because again, both muscular, protein growth, and as well as nerve growth is going to be inhibited. So the three we're going to have a look at is Zellweger's syndrome and X-linked adrenoleukodystrophy, and then later one called Refsum's disease, which is an illustration of the inability to do alpha oxidation. Actually, no. Sorry, my mistake. You rely on alpha oxidation to give you your metabolism of fatty acids. We'll have a look at that a little later on, as I indicated there. But also, we have some issues in regards to the peroxisomes, in regards to utilizing these particular systems to allow us to have fatty acid metabolism. So if we look at plants and animals, there is some differences, and we again can manipulate these particular glyoxysomes and peroxisomes in plants as a model to indicate what is occurring within the normal chain of oxidation of fatty acids. So with fatty acid oxidation in animals, we have the mitochondrial matrix. In the plants, they utilize this peroxisomal system, a organelle, specifically in the process of being able to produce acetyl-CoA from the process of degradation of fatty acids. So let's have a quick look at the differences and the similarities in regards to these two particular systems in the comparison of the mitochondrial and peroxisome and glycosome. And we've already studied the glycosome a little bit already in regards to how we get the interface of the glyoxysome through glyoxylate and the production of succinate to feed into the TCA cycle. So go back a couple of lectures, we actually process it. But all I showed you there was the fatty acids directly producing acetyl-CoA. Now we have this issue where we can understand how that acetyl-CoA is now being produced by an enzyme system of a catalase, where basically we're circulating FADH and FADH2 and FADH with an enzyme to deposit, importantly, those electrons into hydrogen peroxide. So that's why the peroxisome or the peroxide component is there. So we're basically producing hydrogen peroxide in plants, but this is actually then degrading down to molecular water and oxygen in that particular system. Because remember, this is a highly dangerous species, as it's basically a basic material. But let's have a look what's going on in regards to this, where we have a long-chain fatty acid in the mitochondria. We already established this in regards to the FADH2, the formation of the double bond, the hydration, and then the dehydration in regards to forming the ketone, and then the coenzyme coming in with the thiolase to cleave that particular fatty acid off and give us the remaining constituent plus acetyl-CoA. Now, in this case, with the mitochondria, that acetyl-CoA is mostly going to the citric acid cycle, the TCA cycle. But in the peroxisome and the glyoxysomes, they're going to be exported to be utilized in the glyoxysome, obviously feeding those two moieties of acetyl-CoA into that modified system to produce glyoxylate acid and allow them to produce succinate acid, which is drawn off and fed into the TCA cycle. But also this provides, with the peroxisomes and the glyoxysomes, this source of capacity to move acetyl-CoA out of this particular organelle into the cytosol of the plant cell to actually then do work, whether it's producing additional constituents, providing glucose. Because remember, in plants, glucose is a major source for structural elements within the cell through the process of production of cellulose, and therefore we need to utilize a high level of production of glucose to give you those polymeric components to give you the cellulose construction, and that's going to come from acetyl-CoA through the metabolism of fatty acids. So again, we see this in seeds in regards to a high source of fatty acids there, which are going to then ultimately provide us the acetyl-CoA to feed into the phosphoenol pyruvate production, to allow us then ideally to feed the issue of producing glucose as a form of structural component within a cell of a plant. So with plant peroxisomes, there is a similar capacity to the mitochondria. We have a dehydration step, an addition of oxygen by attacking that particular double bond, as we've seen previously, the oxidation of the beta-hydroxyacyl-CoA to the ketone CoA, and then ideally the final thiolytic cleavage in regards to removing the 2-carbon constituent with the CoA, and then substituting a new CoA onto a new carbon number 1 position of the shortened chain fatty acid. So let's have a quick look. That catalase is going through. The interesting thing here, this particular enzyme has the capacity to actually produce heat. So when we look at plants, they're not stone cold. They're actually producing small quantities of heat. And in some cases, some plants that basically utilize their germination in winter actually use enormous amount of heat to allow you to have the solubility of the fatty acids. So I think, it's no good you being a seed in a frozen tundra because your fatty acids will be basically frozen. So you're going to have to generate some heat to keep them in a liquid form to allow them to be moved around, and as well as providing that particular heat energy, lowering those energy barriers and interacting with the various enzymes to do their particular job. In this case, we see a little difference here when we're going from the alcohol to the ketone in regards to the utilization of the NADH. Now, the NADH in this particular case, in the NADH is actually exported for reoxidation from the peroxisome or from the glyoxylate. So we have this capacity to produce something which is not recycled around in the respiratory chain and in making metabolic water, as we see in the mitochondria, as we'll look at this a little bit later on. We don't get that additional component in the peroxisome and the glucosome of making additional ATP. So again, that's a really big difference between the peroxisomes and the glucosomes. They're seen more as a metabolic process in changing the structure to allow you to produce acetyl-CoA without the production of ATP or energy, where in the mitochondria, we have these capacities to produce the NADH and the FADH2 and not recycling it, but moving it out to then feed the system to make ATP. So again, that's a really clear comparative difference between the mitochondria and the peroxisomes and glyoxysomes. So let's move forward. Okay. So in the peroxisome system, because a lot of plant have very more complex and longer fatty chain acids, they have to have the appropriate enzymes to catabolize these long chain fatty acids. And they're usually about above 26 carbons, and they're in their saturated form, because the overall system, as we move down that particular chain of events, has a preference to be able to produce and utilize more shorter chain fatty acid products. Now, one of the problems is some of these particular long chain fatty acids. Some people in the world, particular populations in Scandinavia, have problems actually metabolizing these long chain fatty acids. Ourselves, if you've got a fatty acid disorder, you might have one of these fatty acid pathologies, where basically you're going to see an accumulation of these types of pristinic and phthalic acids and this hexacosanoic acid constituent in your circulatory system and deposited in certain tissues. And that's one of the first things we look for in regards to the types of fatty acids that are present, which gives us an indication of what part of the metabolic pathway is problematic, because these particular fatty acids are being accumulated and can no further be broken down to smaller chain fatty acids that would normally enter in the beta oxidational step. So these long chain active or present fatty acids are usually found in dairy products, fat of ruminants, and particularly meat and fish in specifically in regards to some of these long chain fatty acids and phthalic acids specifically, with some particular types of fish oil. So we've got to be very careful of being able to understand very quickly in regards to what's occurring in the process of beta oxidation, or in this case, how we can help ourselves. If we don't have the full form of beta oxidation, we can still access these fatty acids by using different fatty acid degradational processes. So with Zellweger syndrome, we have peroxisomes in our own body that have this capacity to provide us acetyl-CoA, but in these particular syndrome, we have the inability to make these particular organelles. And therefore, we have the lack of their particular chemistry to handle these long-chain fatty acids. Sadly, there is no present cure for this particular syndrome, and there is actually even no course of treatment, because one thing that this syndrome does is we actually make long-chain fatty acids ourselves. So one of the problems is, if we can't metabolize the natural fatty acid, long-chain fatty acids that are coming in our body through the dietary means, we've got a problem, because we're going to be making our own long-chain fatty acids, which we can't metabolize. So we get this accumulation of these long chain fatty acids. So with most familiar diseases, one of the first things we do as pathologists, when we found a situation in regards to somebody who has a metabolic disorder, we look at how the dietary intake can basically be mitigated or minimized to supplement that particular compound that might be a problem. So in this case, we look at the fatty acid content of their diet and try and mitigate and minimize food that's going to contain those long chain fatty acids. So, as I said, there's no course of treatment for this particular syndrome. And one of the problems is with some of these long chain fatty acids, they actually provide a process in membranes. And without those particular membrane fatty acids being present in stability and weakness of those particular membranes, there's a higher level of susceptibility to infection, and that in particularly of pneumonia and what they call respiratory distress syndrome. So we got this horrible other issue occurring now with infections, given the abundance of these long chain fatty acids. And basically, we have to then provide, respiratory ventilation, antibiotics. But again, the issue is it's pretty poor prognosis. Death usually occurs within 6 months of first observing the long chain fatty acids. And there's a whole lot of complications in regards to basically the body starts shutting down. We see enormous amount of weakening of the membranes, which induce gastrointestinal bleeding. The liver starts to fail because of, again, the incapacity to metabolize those fatty acids, because what we see is what we think called fatty liver, where there's a very large deposit of fats in the liver and causes then a much larger size of the liver to basically increase its size maybe one or two times over. So that's a very, very serious particular disease, and that's again because we're not being able to produce the peroxisome organelles to then also allow the transportation of those fatty acids, those long chain fatty acids, to be degraded down to smaller chain or to acetyl CoA. Now, the other one, the X-linked adrenoleukodystrophy, or called XLD, ALD. Again, this is the issue directed specifically at the transport system. So again, we get the accumulation of those long chain fatty acids, the C-carbon 26, and not being able to have those brought into the mitochondria, into the peroxisome, and then be broken down to be smaller fatty acids to then be handled by the carotene transferase number 1 to move those into the mitochondria. And so we get this accumulation of these long chain fatty acids. Now, it's an X-linked, so there is actually a genetic deposition for males in this particular component. And one of the things that we see commonly is loss of vision because of the optic nerve and the demyelation of the optic nerve specifically, which is a nerve that goes to the brain and to the eye. We also see usually mental retardation and behavioral disorders, and sadly, death within a few years of analysis. So most of the kids, the boys, they die before the age of 10, and it's not a very nice disease to actually witness. So we know a lot about this particular one in regards to the mutations that have occurred. So CRISPR might be a approach to be able to utilize genetic technology to correct the ALD protein, which is the transporter in regards to moving that particular fatty acid into the peroxisome. So this is a protein that is a transmembrane protein. It belongs to a family of proteins called the ATP binding proteins, and it's involved in moving those very long chain carbon fatty acids into the peroxisome for that process. And what we see commonly as I said, I'll illustrate here. So here is a normal nerve in regards to the axon and um, nodules, what are they called? I can't remember their name now. Nodules, not neurons. They've got nodes. Nodes. That's it. They're called nodes. And so what we see here with somebody with a particular disease state in regards to the demyelation, there's the nerve. Here's the insulation, the myelin around the thing with the various little cusping of the nodes. And look at, it's just basically an open tether, which is not good. We also see through biopsies in regards to the adrenal cortex, this type of stuff in regards to very high deposits of these long chain fatty acids present in the tissues. So again, they're illustrating clearly that there is an inability to metabolize these, and they're being stored in various organelles in regards to being able to produce these types of sectioning and illustration of the accumulation of those long chain fatty acids and clustering within the various tissues, which is going to stress those particular tissues. So in plants, and this is actually a very important illustration in regards to what we see occurring in the capacity of utilizing fatty acids, not just as a energy source, where in plants they become a very central part from beta oxidation as plant precursor constituents. So fatty acid oxidation in plants, we see the peroxisomes and the glyoxysomes being an important role, and we've talked about peroxisomes in regards to the catalase and producing producing amounts of acetyl-CoA by digesting and degrading large long-chain fatty acids. But we also have that glyoxysomes utilizing that acetyl-CoA, now giving you enormous amounts of more energy from the lipid body and the fatty acids. So this is what we're talking about here, this transition from fatty acids to acetyl-CoA. And here we have our friend the glyoxylate cycle producing succinate and consuming two moieties of acetyl-CoA and cyclating around and allowing us that succinate to actually be transported out, to be fed into the TCA cycle, to then go through malate or oxaloacetate, and to go through the malate shuttle in this case, illustrating the regeneration of oxaloacetate, and remember the reducing equivalents on the cytosol, going through phosphoenol pyruvate, and then going through all the process of being able to produce sucrose. So when we think about our triacylglycerols in the seeds with those lipases, we are taking that triacylglycerol or diacylglycerol, we're taking that fatty acid, we're going to bring that into beta oxidation. But remember, we are also contending with the glycerol backbone to make further energy to allow us to utilize that as glycerol 1-phosphate and glycerol 3-phosphate, then glyceraldehyde 3-phosphate. So we're metabolizing every little piece of those fatty acids or those triacylglycerols to provide that particular process of energy in plants. So through beta oxidation, we have that acetyl-CoA coming in, which is then going to either feed the TCA cycle, or in this case, we're going to feed the glyoxylate cycle in regards to getting more of a constituent in the form of succinate to produce those particular sugars. But also through the glyoxylate cycle, we can go and produce oxaloacetate and feed the process of gluconeogenesis. So we've got a couple of pathways we can pursue in plants to really supplement the glucose level, because remember, as I said before, glucose is such an important aspect in plants in regards to both their metabolism as well as their structure. So with the glucose constituents and the high pool of glucose we see in plants, we know we can make various nucleotides in regards to making those furan sugars. We can take those glucose off to make amino acids. We can take the glucose directly to making energy, and glucose making polysaccharides for structural components. So again, in plants, glucose becomes a very, very important commodity in regards to allowing them to survive and to actually grow and develop. And again, you can actually learn more about plant biochemistry if you want to do our graduate plant biochemistry course, which refreshes your memory on all of this stuff, but goes into a little more detail. You've got the Calvin cycle, you've got a lot more on photosynthesis and plant hormones. So there's some very unique aspects from the different from the mammalian system in regards to plants, and you can learn more about that if you so desire in our one of our graduate classes, run by the other biochemist, Dr. Owens. So one thing we're very interested in is in how all these systems of regulation, as well as the enzymes involved in these metabolic pathways, have evolved and even diversified. And we see these examples in regards to the peroxisomes and the mitochondrial systems in regards to substrate channeling, how these enzymes have come together in regards to both structural and association. So structural is where we might have a Janus molecule of multiple faces doing separate components, allowing substrate channeling to be more effective. Or we have a number of individual proteins associated with a lipid raft or a membrane coming together to be able to do an appropriate and efficient movement of a substrate product interaction through a chain of commands and events. So with gram-positive bacteria in their mitochondrial system, they have the capacity to only deal with short-chain fatty acids. And they have this capacity to produce an enzyme, take that substrate to produce a product, which is an intermediate, which will then go to the next enzyme. And you can see there's a requirement for diffusion in regards to producing the final particular product. Now, with gram-negative bacteria, this association has become a little bit more complex and a little bit more efficient in regards to the first evolvement of enzyme 1 and 2 coming to actually together on a Janus molecule and then interacting with enzyme 1 and enzyme 4 to do their job with a more sufficient way of basically a channel substrating of illustration of channeling the substrate through the individual enzyme system instead of allowing these intermediates to diffuse freely within the cytosol of the gram-positive bacteria. So again, the time efficiency for a molecule from here to diffuse from enzyme number 1 to 2 to 3 to 4, it takes a much longer process compared to the channel substrating situation we see in gram-negative. In mitochondria, where we're looking at very long-chain fatty acid specificity, again, we have membrane-bound particular enzymes in the inner membrane of the mitochondrial matrix. So this is facing the inside of the mitochondria. So we have another membrane here, the outer membrane matrix, and the inner membrane component between the two mitochondrial membranes. So we're dealing with now the fatty acids coming in, and with the enzymes 2, 3, and 4 being actually highly associated with each other due to the lipid raft and due to association within the lipid membrane, giving us a higher capacity of transferring the substrate and the intermediates to produce the final product. So again, a lot more efficiency. And then in the peroxisomes and glycosome systems, we see a more further development in regards to a multi-Janus molecule, in regards to having multiple enzyme sites being present, as well as an association with the first one, the enzyme complex, to give us a more efficient route and maybe even a diversificational route in regards to what we do with that intermediate. But we still, in the peroxisomes and glycosomes, we still have this other component where there's going to be a slower intermediate release, which is going to have to diffuse to the enzyme number 4 to finalize the product, in regards to, in this case, must be acetyl-CoA. So again, by studying these types of proteins, we can start realizing how things have diverged and or a point of origin where really, this is the point of origin, which we will normally think about enzymes in a cytosol producing a product, an intermediate, which then has to diffuse to the next enzyme to then be actually regulated and then go to the next product, etc., to finally get to, the final stage. But as we see through the process of evolution, there's become a higher level of efficiency in regards to bringing these enzymes in close proximity or even associated Janus molecules, which then gives the capacity to indicate their capacity to regulate and process these particular fatty acids. So I've gone through these particular components, and they all have the same enzyme kind of configuration to what we're seeing with beta oxidation. The hydrolase, so the dehydrolase, so the dehydration, the hydration, the rehydration, and the thiol ester or the addition or the thiolase cleavage to give us then the final product of acetyl-CoA. So again, we're utilizing a slower system because we have intermediate diffusion between the enzyme systems. With the gram-negative, we've seen the two parts of the enzyme becoming a Janus molecule. So we have an active site here and an active site within the same protein, and passing specifically through channel substrate channeling, passing that product from enzyme number 2 to enzyme number 3 and an associated protein. So enzyme number 2 and enzyme number 4 are associated proteins, which might have a high attractive association, mostly ionic and hydrophobic interactions with the Janus molecule. Okay, and as I said, this is more typical to what we see in our own mitochondria, where we have a Janus molecule, a two-faced surface molecule, but the orientation and the approximation and movability is really kind of attuned to the issue of embedment in the lipid membrane of the inner membrane of the mitochondrial matrix. And then this one is most probably one of the best systems, but again, it means that enzyme 6 and 7 might have the capacity to be regulated by other things, which would not normally be a part of the regulation of this system. Because there is a drawback. When you have these multiple enzyme sites, you are opening yourself for more regulation. If you're doing some kind of enzymatic regulation on enzyme number 5 and using allosteric modulator, it might inadvertently affect enzyme number 2 and enzyme number 3. That could be a good or bad thing. But again, we've got to be very, very careful in regards to where we regulate and control the system, because we might be over-regulating the system in regards to attacking or modulating one of these erogenous or one of these distant enzyme systems that are still a part of the main protein, which has now 4 individual facets to enzyme activity. This is mostly also a problem because the larger size of the protein has a higher increased possibility of allowing to produce mutants. And again, if we have a mutant in enzyme number 6, the structure of the whole protein might be detrimental in regards to its inhibition of acting as enzyme number 2 and enzyme number 3. So we've got to be very, very careful in regards to how we interpret and how we utilize these larger enzyme systems of these multiple facet sites because they are more susceptible and more problematic in both mutation and regulation, and might have a whole impact in our larger viewpoint of the system. So I want to now look and switch modes to another fatty acid degradational process. Now, what happens when we start seeing defects in beta oxidation, we can survive to a degree by now promoting an alternative pathway. And that alternative pathway is what we call omega oxidation. So if we think about our nomenclature for our fatty acid naming, we have carbons number 1, 2, 3, we have alpha and beta carbon in regards to the carboxylic acid end. But on the far end, the end furthest away from the carboxylic acid, we have the omega carbon. And what we basically do there is we go through the same process as what we're seeing with some of the other substrates in beta oxidation and promote now the production of a hydroxylation on the omega carbon, and then go through the process of making an aldehyde, then that aldehyde making it to a carboxylic acid, and then using the enzymes to attack that carboxylic acid side that we've done in the same way in regards to the carbon number 1 in the normal beta oxidation process, and attacking the omega carbon from the other side of the fatty acid molecule, and going through and removing 2 individual carbon moieties at a time in the form of acetyl-CoA. So let's have a look at that. Geez, I'm doing too much talking. So what we see with beta oxidation, we're degrading usually at this particular site, the carbon number 1 position. And with omega oxidation, we're oxidizing at the most distal carbon from the carboxylic acid. So this is usually a minor pathway in the normal healthy individual in regards to beta oxidation. But what happens when we have a situation of defect, when we want to start metabolizing these short chain fatty acids to get them to become carbon 12 and carbon 14 fatty acids, because these are the guys that are the best substrates for the liver and the kidney. So what we actually do is use a mixed function oxidase utilizing NADPH and oxygen to provide that alcohol constituent on that omega carbon. Once we have that particular omega carbon hydroxylated, we then utilize NADH, or say NAD plus, and make NADH through the alcohol dehydrogenase enzyme to give us that aldehyde constituent. So we now have a carboxylic acid and an aldehyde at that omega position. Once we have that omega position with the aldehyde, we can then go through a further step utilizing the aldehyde dehydrogenase. So we're using an alcohol dehydrogenase, then an aldehyde dehydrogenase, sinking those additional electrons and providing that proton and removing that proton to give us the carboxylic acid. So we now have two kind of carboxylic acids with a chain, an alkyl chain, and it could be saturated or unsaturated. And so what we can do is then go through the normal stages of beta oxidation. And in this case, what we're doing, instead of removing acetyl-CoA, we're going to be reducing succinate through beta oxidation, because we're going to do the same process and we're going to be reducing succinate, which is a constituent within the TCA cycle. So we can feed this omega oxidational product directly into the TCA cycle by producing succinate. So again, we're going to see a number of carbons being taken off at a time. So in this case, we see succinate, which is 1, 2, 3, 4 carbons, not a 3-carbon constituent, not a 2-carbon constituent like we normally see with acetyl-CoA, but a 4-carbon constituent coming off as a form of succinate. We also see this apatate molecule, which is going to be a residing molecule in regard to now having to deal with this particular compound, and we're going to do that efficiently by actually degrading it further to make some moieties that we are previously familiar with. So what we see again, sorry, that was actually a mistake. There's supposed to be another slide there. Okay, well, this further gets broken down to a 2-carbon constituent involving acetyl-CoA, but I think there's a slide missing, unless there's a slide out of order. So let's have a look at another fatty acid degradational system. So we've looked at omega. So omega is that issue with that omega carbon going to an alcohol, to an aldehyde, to a carboxylic acid, a cleavage, giving you the capacity to produce succinate to feed the TCA cycle, and the resulting product, which is the remainder part of the fatty acid, will slowly be degraded to acetyl-CoA. Now, phthalic acid is an interesting fatty acid where it has branches at various carbon positions along the carbon backbone. And we've got to be able to deal with these by basically promoting the process of a hydroxylation. Now, this particular system gives us the capacity to basically skip to a part of beta oxidation with that hydroxylation, and allowing us basically then to form that double bond, form the ketone, and then go through the thiolysis that we've seen previously in the beta oxidational process. But there's a couple of extra steps in regards to being able to produce that particular constituent through hydroxylation, decarboxylation, aldehyde, and then the oxidation to form the carboxylic acid. So let's have a look at what we see with phthalic acid. So here we have the branched carbon in the carbon number, so carbon number 1, number 2, number 3, the number 3 position. So this is the alpha position, here is the beta position. And we're going to go through and swap this over in regards to allowing us to utilize coenzyme A to get the carboxylic acid primed for the enzyme system in regards to consuming ATP to promote that particular thiol ester reaction that we've seen previously in all our fatty acid carboxylic acids going to produce that coenzyme priming by the consumption of CoAsh and ATP. With that CoA component, phenol CoA, now being present, we're going to use the phenol CoA hydroxylase and provide us that first step in the form of hydroxylation. With that first form of hydroxylation, we're going to target this particular bond in regards to allowing us to put that hydroxyl component. So we're utilizing ascorbate, alpha ketoglutarate, and iron, and producing succinate and CoA as a derivative. So we're basically utilizing alpha ketoglutarate as an electron sink and bond sink in regards to breaking even alpha ketoglutarate up a little further to produce succinate. So we produce that hydroxyl position in the carbon number 2 position, and then we're going to then actually go one step further and deal with that particular hydroxyl position in the formulation of now leaving that formal group in as a good leaving group and giving us now a reductional process in the process of decarboxylation to basically allow us to have this aldehyde in the form of pinosyl fatty acid. With that aldehyde being present, we're going to then go through the aldehyde dehydrogenase, consuming either NAD or NADP to produce NADH or NADPH, which will give us then that carboxylic acid. That carboxylic acid then is going to give us the whole process to basically jump that particular bond positioning and be able to look at this alpha and beta carbon as our target constituent to be able to cause that thiolysis reaction and get this guy off and then shorten the chain length of the branched chain fatty acid. So we'll go to the next one where we have the pristinic acid and going through the normal stages of beta oxidation. But in this case, formulating now a constituent of 1, 2, 3 carbons, not 2 carbons in the form of acetyl-CoA, but formulation of propanol-CoA, which we all know can go through the methyl-manyl-CoA and allow us to form succinate to feed the TCA cycle. So again, we have this capacity to move these through the similar situations of beta oxidation and allow us to actually get to this guy in regards to forming, so there's carbon number 1, carbon number 2, and carbon number 3, the alpha-beta bond being there, and going through the normal forms of beta oxidation to produce the propanol-CoA through the thiolysis, and the regeneration of a shortened chain of branched fatty acid, which will then also go through the same process all the way back at the beginning of here and going through the hydroxylation again and decarboxylation, aldehyde, and carboxylic acid formation, as we've illustrated here, to again give another section of 3 carbon constituent in the form of propanol-CoA coming off in the thiolysis reaction. And again, as I showed here, with the propanol-CoA and the biotin propanol-CoA carboxylase dependent enzyme interaction with bicarbonate and ATP, we're going to get the methyl-manyl-CoA, and then we're going to actually go from D to L form to produce then succinyl-CoA with the methyl-manyl-CoA mutase, and then feed that succinyl-CoA to feed the TCA cycle to produce succinate, and then feed the remaining component of the TCA cycle, but importantly producing an equivalent of GTP. So again, we have this little side step which we can utilize with those branched chain beta fatty acids such as photanic acid and give us that capacity to again feed the TCA cycle. Now again, this may not be an apparent metabolic system if you have a defect such as in Refsum's disease. Now, Refsum's disease, we look at the phenol-CoA hydroxylase. So let's go back. Where are we? Phenol-CoA beta. Yes, this guy here. where what we will see is an inhibition of this particular enzyme having the inability to take the branched fatty acid to become hydroxylated. Therefore, we are not going to proceed to the production and shorting of the fatty acid chain to make the succinyl CoA. We're going to see an accumulation of the fentanyl CoA being present in the circulatory system and in the tissues. This particular disease, can you notice something about the individual's feet? Again, we see this is actually an extremely characteristical component of this particular disease. The mutation of the phalanges, or it's the metatarsals, the toes. And so what happens here, photanic acid is very high in concentration and basically promotes deafness and blindness, again through that issue of affecting, in this case, both the auditory nerve as well as the optic nerve, and basically causing other neurological problems such as mental retardation. Now, the deficiency of this particular photanic acid is usually 100 times more in circulation than normal, and also deposited heavily in the liver and the kidney and the heart. So we can see this by again doing biopsies and seeing this very large cluster of little rosettes of fatty acids being present, and basically doing an analysis on those particular fats and indicating that these particular types of branch-chained fatty acids as the fentanylic CoA component. Now, the main process in regards to controlling this is limiting the dietary intake of photanic acid. So the problem is, as I said before, with some of these metabolic disorders, we also produce these components. So you're fighting a losing battle in regards to not being able to metabolize this fatty acid, but your body is also producing that fatty acid. But it takes a much longer time to produce it and accumulate that to a very high level, which is going to be more dangerous and lethal. So in this case, we tried and find a dietary constituency in regards to minimizing the content of those particular fatty acids present in the diet. Now, so when we look at some of the foods and photanic acids around, so in tuna, lamb, beef, cod, pork, and ham, and interestingly, you do not see this being regulated by the FDA to like we see in the phenylketonuria in our cans of soda with the artificial sweetener. So this is more a reflection of the genetic presence of this disease state in the population within the United States. So the EU regulates this by putting warnings on cans of tuna, and other meats in regards to the types of fats and oils that are present in those particular processed cans or meats. So they provide those particular regulational indicators, where here in the US we don't. So I'm now going to switch phases in regards to the issue of not degrading fatty acids, but what do we do in a biological system when fatty acid metabolism has changed? That we have no longer a source of glucose as well as coming from glycogen. We have a limitation of glucose coming through gluconeogenesis, which we're promoting for that glucose to go to the brain. And then we're still short in regards to the amount of energy that the brain is needing and wanting. And we're going to have to try and basically supplement that system with another commodity to allow it to do its normal process. And the normal process in this case has been met by the process of producing ketone bodies. Now, if you have not had breakfast and you're wearing a mask, you can mostly smell your own breath. And sometimes some breaths smell a bit acetone. So, you know what acetone or nail polish remover smells like a little bit? That stuff is a byproduct, a volatile byproduct that is coming from your lungs through the process of the change in metabolism, producing these particular constituents. And these constituents, in the form of ketone bodies, become a source of energy that is exported around the body. They are blood soluble or liquid soluble, and they have this capacity to basically be circulated from the liver and feed the brain specifically. But there is some consequences. We cannot live on ketone bodies alone because there is some detrimental effects of these particular systems. So let's think about our fatty acids. We've degraded those. We've got no carbohydrates. We're going through gluconeogenesis. We're taking our acetyl-CoA to go through gluconeogenesis. Very expensive procedure. Remember, we're consuming NADH. We're consuming ATP to push that material through gluconeogenesis. It would be better for us to find a deviate path to allow us that acetyl-CoA to feed something where normally it's feeding the TCA cycle, but again, our energy requirements, we're going to have to be feeding some of that acetyl-CoA to keep the NADH levels high. We see the ATP levels need to be high because we're seeing an accumulation of AMP and NAD plus because we've got a low glucose level, low energy level. We are now starting to metabolize our fats to do this particular job. So we're going to form these particular ketone bodies from acetyl-CoA, and it's basically an illustration of enzymatic condensation reactions in regards to producing this constituent in the hydroxymethyl-glutyl-CoA constituent, which will then later become a energy source for the brain. This comes from the process of condensing a number of acetyl-CoA moieties in reversible reactions. So here we have the first condensation reaction where we have a great leaving group in the form of a coenzyme A, and we're going to go through the same thiolysis enzyme that we saw previously in beta oxidation because that's a reversible reaction. But this time we're going to actually release the coenzyme A from the constituency and provide us this capacity to interface with this methyl group and condensate two molecules of acetyl-CoA to produce the acetyl-CoA constituency. So here we have our first acetyl-CoA constituent, our first two carbon constituent, and our second two carbon constituent from the original acetyl-CoA. We are then going to see a third acetyl-CoA come in. So there's three acetyl-CoA constituents coming in through a condensation reaction, first to 2 and then 2 to 3 in regards to using the hydroxymethyl-butyrate coenzyme synthase, which is going to consume acetyl-CoA and water, release the CoAsh, and give us now a sticky end on the other part of this guy by reducing this ketone to become an alcohol, and then put the carboxylic acid of the remaining component of the acetyl-CoA onto that particular central carbon. So we have like a symmetrical molecule where we basically have a coenzyme side with a ethyl ester, with a side with a carboxylic acid. We have a carbon either side of the central carbon, which contains the hydroxyl constituent. With that process, we can then go to the modification of the hydroxymethyl-glutyl-CoA through the MG, so the hydroxymethyl-glutyl coenzyme lysase, which gives us the first capacity to release a moiety of coenzyme A and in the form of acetyl-CoA. So with that, we release two carbon constituents, which can then basically produce energy and be fed into a system such as in the brain. We then produce the remaining constituent in reform to acetoacetate. Acetoacetate, which is a very interesting compound because it can actually be further metabolized to the liver, specifically the liver. There's an enzyme that's only located in the liver to produce the acetoacetate, which is now blood soluble and can be circulated in the system. So it's going through the circulatory system, it's going to pass the liver, and it's going to then produce this moiety in the form of beta-hydroxybutyrate. So again, it utilizes the NADH and a proton system to deposit those particular protons into the acetoacetate and allow us then to basically sink those electrons to produce the alcohol constituency in regards to the formation of beta-hydroxybutyrate. There is a other enzyme system, the acetoacetate decarboxylase enzyme, which causes the release of carbon dioxide from this constituent in the form here releasing this guy as carbon dioxide gives us then the form of acetone. That form of acetone is the one that we see in regards to the smell of an acetone breath. So a person who is going through a metabolic process where they're utilizing this particular system in regards to providing fuel to their brain is actually producing acetone, which can be smelt in the breath. And some of these new diabetic machines to indicate, well, in the old days, you used to be able to prick your finger, you do analysis of the glucose level in circulation in your little finger, and that gives you then basically how much insulin you need to inject yourself with to combat that increase in glucose in circulation. Now they use this system where they actually measure the acetone in the breath. And by actually measuring that acetone in the breath, they can actually determine how to combat this particular system in regards to trying to increase the carbohydrate level into the cell by providing you the amount of insulin that you need to inject to get that system going. Because at the moment, your body is not getting that carbohydrate into the cell because the insulin receptor is not working. The GLUT transporter system is not working because the insulin receptor is not working, because there's no stimulation of insulin, because either insulin secretion is not being undertaken by the pancreas. So in this case, what you're trying to do is stimulate the whole system to say, okay, let's switch the metabolic process from these ketone bodies back to the normal glucolytic system. And that's what we're trying to do when we're trying to managing diabetes. But in this case, what we can utilize this acetone breath in the form of the exhalation to give us an indication of how far we are with this ketone body production. And as we administer an amount of glucose, and this is set by what body weight mass index and all these other things, there's a table you kind of look at how much acetone you're producing. So if you give yourself this much insulin, you should be able to combat the amount of acetone you're producing to then move the metabolism from ketolysis or from ketone body production to basically glycolytic production in regards to utilizing your glucose stores and utilizing your glucose in the circulatory system. So again, it's a less inhibitive in regards to getting that finger prick. You just basically breathe into this device and it gives you a number. So with this particular guy in the normal process, what we deal with as normal individuals without diabetes is we're having this effect occur when we are in the starvational process. So we've got no glucose, we're metabolizing our fats, we're producing acetyl-CoA, as well as we might be in a starvational situation. If we even go further, where we start metabolizing our proteins to produce more acetyl-CoA, we're going to have to get that acetyl-CoA into the circulatory system somehow. It's not soluble as its normal form. We're going to have to condense it by using the three moieties of acetyl-CoA, producing the hydroxymethyl-glutyl-CoA, and then the acetyl-acetyl-CoA, which is soluble to go to the brain, but has to pass the liver to produce the beta-hydroxybutyrate, which is going to be the key constituency in regards to the delivery of that molecule to the brain. So let's have a look. So beta hydroxybutyrate as a fuel system. I'm actually going to stop it here because this is actually an important step that I want you to kind of understand and know. And we'll talk about it when we talk about this on tomorrow. And then I'll give you some more illustrations and how this all connects specifically in regards to what we understand in regards to beta oxidation, ketone body formation, and their utilization as an energy source. So let's stop it there because if I go too further, I'm not going to run out of time. And I know some of you people have other places to go to during that time. So I'm going to stop it here. So I will see you tomorrow. We'll have a discussion in office hours in regards to this material we talked about yesterday and today, because yesterday's material was actually a heavy load. You're going to have to really understand those processes of beta oxidation and now reflect on our different forms of alpha and omega oxidation and how we utilize, again, the same processes in beta oxidation. Because basically what we're doing, we're modifying our individual substrates to form moieties that are similar to substrates that are in the normal beta oxidational process and substitute those in to get us the capacity to produce 3 carbon constituents such as propanol-CoA, or 2 carbon constituents in the form of acetyl-CoA, which we're now starting to understand in regards to why we want to use that acetyl-CoA as an energy source to make it soluble through the beta-hydroxybutyrate. So we'll talk more about that tomorrow, okay? I keep running out of time, guys. to talk to you all this stuff. So see you tomorrow. So yeah, it is 9:20. Yep.